Hello, Gut Check Project fans. Welcome to Gut Check Project and KBMD Health Family. I'm Eric Rieger here with my awesome co-host, Dr. Kenneth Brown. We have another special show.
Holy cow, we just keep outdoing ourselves with smart people. I'm becoming very comfortable being the stupidest person on these Zoom calls right now. This is the new norm. Me being the absolute dumbest person on this screen right now.
Thank you. That's really weird. And if you're the dumbest, then this is going to be a really, really intelligent show. So today, episode number 52, we have a special guest. This is Dr. Charlene Van Buiten. She is an assistant professor of food science and human nutrition at Colorado State University. Hello, Charlene. How are you doing today?
I'm doing well. How are you guys?
We're doing great. I'm not going to introduce the paperwork that we're going to get into, but what I am interested, before we get into some incredible information about your research around celiac disease and how essentially people can stave off long-term inflammation. We always like to get to know a little bit about you, but we did print off your resume, and it looks to me like from all of the stuff that you do in this CV, you started sometime in the womb, getting things done.
So you've been like publishing papers in utero somehow?
Yeah. It's a really rare skill.
So where are you from originally?
Originally, I'm from Connecticut. Grew up there, and then did my bachelor's degree at University of Connecticut in nutrition.
Nice. And how long have you been at Colorado State?
I've been at Colorado State about 1.5 years now. So I got about 6 good months before everything shut down. Still happy to be here. It's a nice place to be locked down in, at least.
Oh, yeah. Absolutely. Northern part of Colorado. It's definitely beautiful up there. So before you were at Colorado State, let's see, where else? You were a postdoctoral fellow, obviously, and then is there any other stops along the way that really leapt out to you and ended up pushing you into Colorado State to do, food and nutrition for human?
Yeah, I would say probably the most important step in my whole academic journey was at Penn State, where I did my PhD in food science. That's kind of where all of the research that we'll talk about today was really conceived. And it was just kind of a one-off idea that my PhD advisor and I had just one day, what if we looked at interactions between gluten and tannins? And then, I kind of went back to my office, first year PhD student, real excited. And all of a sudden it was like all of these ideas just in terms of the chemical interactions and what this means for nutrition. It just designed this whole project. And in a really rare case of circumstances, everything I had designed in my first year of my PhD ended up being what I eventually did over the course of the next 5 years. I don't think anybody is ever that lucky. So, and the project turned into what you all were able to read before inviting me here.
Awesome. Well, we will definitely dive deep into that, but I'm really curious. So we have the CV here that shows your academic pursuit. I want to know why Dr. Charlene decided to do the study of food science and nutrition. Let's just start from there, and then I get the passion once you latched onto something, but I'm always curious how people find their way, like how you got there.
Yeah, I think I discovered food science a little earlier than the average person. Most people will get into it in college, having followed a path of chemistry or biochemistry, and then realizing they can apply all of these concepts to food. But I actually was in the Future Farmers of America when I was in high school.
Nice.
And I thought that I wanted to be a vet, and then realized that I was not really into blood or sick animals or anything. And one of my teachers was like, we have this competition. It's called food science. You design a food product. You talk about safety and everything. And I was like, sure, sounds cool. And the first day that I met with that team for this competition, we got a textbook chapter on canning. And I was reading about canning, and I was like, oh, this is really interesting. Like, if you can something, it'll last a really long time. Or if it's done incorrectly, it can be so dangerous that it can kill you. And that kind of dichotomy there, I was like, food science is crazy. And just from there was like obsessed with it. And
Awesome.
yeah,
That's
then
really
went to
cool.
college for nutrition, grad school for food science. So.
This kind of reminds me of the whole mycology thing that we were talking about before, where like you might find a brave food canner and you might find an old food canner, but you won't find them in the same person. It's like, I'm going to just dabble in some aggressive food canning and see what happens. They don't walk around anymore. We had Paul Stamets was talking about that. And then, the other mycologist, we've talked to mycologists that actually discuss that. They feel the same way, where it's like, look, you can have a mushroom that'll save you, but if you prepare it wrong or eat the wrong one, you're going to die. If you have food canning, you can have food forever, but if you do it wrong, you can die.
Yeah.
Well, that is awesome. So reading your article or reading your paper, which is a review of your thesis, which tells me that you know this essentially better than anybody in the entire world, because you did a thesis on something that I have been searching for a very, very, very long time. I developed Atrantil to help people with bloating and irritable bowel syndrome, and then we started learning, started learning about these effects of polyphenols. Came across your article, your review, which is titled Gliadin Sequestration as a Novel Therapy for Celiac Disease, a Prospective Application for Polyphenols. This is the thing that really I've been looking for a long time. We've known the benefits of polyphenols, but you're the first person that has been able to explain why I'm gluten sensitive. And once I started taking Atrantil, whenever I would eat gluten, I didn't have issues, and I really couldn't explain it. We've had patients that say, when I take these large polyphenolic compounds that are in Atrantil, I can eat wheat. Why is it? I didn't know. I couldn't actually say from a molecular reason. And then I came across your paper, and this is absolutely fantastic. It's 32 pages of incredible material and 185 references. My goodness, you put some work into this. Congratulations on putting together what I think is the most comprehensive review of polyphenols in the setting of celiac disease. So once again, if anybody listening to this knows somebody that has celiac disease or has a family member or has celiac themselves, this is something that really we need to share as a community. We need to get this out there, and your work is really pivotal to explain the science, which is so cool. So let's jump into it because it's awesome.
Yeah, I don't even know where to start because you described multiple different mechanisms of action on why polyphenols begin to work. So what drove you to put these two associations together? Why celiac disease and why polyphenols?
So at the time that we came up with this project, I had recently joined my graduate advisor, Ryan Elias at Penn State. I had joined his lab, and he was doing a lot of work on wine quality. So as a food chemist, we're looking at oxidation of polyphenols, how that can affect wine astringency, et cetera. And so I was, showed up to grad school thinking I was going to work on wine. And then we found these papers that were studying protein polyphenol interactions using tannic acid with peanut allergens. And so that was an interesting paper, doing a little bit more reading, thinking about that, and then thinking about, still wine. And I came across these papers that were using gluten as a fining agent in red wine. So fining is the interaction between polyphenols in wine and a protein that will actually precipitate those polyphenols and take them out of wine to kind of soften the mouthfeel. And so I saw that, and knowing that gluten is this immunostimulatory protein, I was like, why are they doing this? That seems crazy to me. And is it hurting people? And, it's not labeled because it's something that's not technically in the product because it's falling out as a solid.
Sure.
And that was just sort of the end of the string on that ball of yarn that we kind of started to unravel. And looking at that in terms of a food processing aid, I thought, what if we looked at this from the perspective of a nutraceutical? So if we know these interactions are happening already, can we flip this around and put it in the human body? Can these interactions still occur? What is happening to the protein? How does that affect the actual mechanism of the disease? And, we just came up with all of these questions from there, but it really started with gluten as a fining agent in wine.
Okay.
That's incredible. That is a lot to peel back as you're starting your sort of academic career to take this on and then suddenly go down a rabbit hole and you end up over here with a disease, celiac.
Right.
That is, and thank you. I mean, that is crazy to take on that. You just discussed layers and layers. I imagine you were looking at an article that then led to another one, and then you finally went, oh, this is way bigger than I thought.
Yeah, those 185 references on the paper is just like the tip of the iceberg.
Well, hey, just to reset for everyone too, those who suffer from celiac disease obviously know about gluten and why to avoid gluten, but maybe not everyone understands why gliadin specifically is what we're concerned with its block. So why don't you explain a little bit about gliadin itself?
Sure. So gluten is a heterogeneous protein made up of two subunits. So we have the gliadin as well as glutenin. And those two proteins will come together, forming inter and intramolecular bonds in order to form this gluten protein kind of as a whole. But it's the gliadin that has these repeat motifs in its amino acid primary sequence. And it's usually a proline that's one amino acid away from a glutamine that's going to get recognized in the human body by an enzyme called tissue transglutaminase or transglutaminase 2. And that gets deamidated. And then that's the area of the protein that's recognized by antigen presenting cells. So it's really comes down to the amino acid sequence in the gliadin versus what's typically seen in glutenin.
So step back, what you just described is exactly what happens to 1% of the population, because celiac disease is the most prevalent autoimmune situation or autoimmune disease that's there. And what you just described was the amino acids, the proline and the glutamine, actually form the gliadin. Am I hearing that right? So those two form the gliadin.
Right. So they're found within the structure of gliadin. So gliadins really are a class of proteins. There's alpha, beta, omega. And then within that, alpha 1, alpha 2, alpha 3, there are so many types of gliadins, but sort of this consistent pattern that we see in those gliadins is a really high percentage of proline as well as glutamine.
Okay.
So in your article, you did describe something that I wanted to clarify on this. You described the prolamine, glutamine residues, and the sulfurous component of prolamine is cysteine, and the ability to form these disulfide bonds. All of that, is that why gluten makes things spongy, because of these disulfide bonds? Is that why it's used in so many things?
Yes. Yeah. So disulfide bonds are formed within the structure of gluten over the course of hydration, oxidation, and mechanical kneading. So that's what you see when you're making like bread or pasta and that dough sort of starts to really come together. That's a result of the formation of disulfide bonds.
Nice.
Which is what makes which it's that texture that which is why when you get gluten-free bread, you're like, no, it's not
right,
the same.
Right, yeah. So it kind of forms that balloon structure through leavening.
Yeah, so I've actually sent back some gluten-free pasta and said, can I have some more disulfide bonds in here?
Yeah, they don't have that in a shaker, Ken. Yeah. That's not there.
That is impressive. So why gliadin? Why gliadin? Because we know that gluten is formed of these two different proteins, but why gliadin being the more problematic protein in the gluten molecule?
So gliadin is more problematic because, as I mentioned, that higher frequency of the amino acid residues, the proline and the glutamine, just found in that particular order, proline, something else, glutamine, is what's going to make it more recognized by the body. It's also this high amount of proline results in almost like an unraveled protein structure. And that is something that's recognized structurally by the body as being a problem. It's more difficult for enzymes to break down gliadin because of all of those prolines, because of that unraveled structure. And then that unraveled structure allows it to be, it allows it to interact with that tissue transglutaminase enzyme as well.
Okay. So let me stop you right there. So you said something, and that's kind of what I wanted you to say, is that gliadins are resistant to digestive enzymes.
Mm-hmm.
So you have this thing that your body can't readily break down. So as a scientist, my patients ask, what's the deal? Why is celiac on the rise? Why is it exponentially on the rise? Why are more people saying that they have gluten intolerance, whether or not comedians will make fun of that, like it's something that's in your head? But I am very gluten intolerant, but I don't have celiac disease. So why do you think we're seeing more of it?
There are quite a few ideas as to why we're seeing more celiac disease. And I know that one of the prevailing hypotheses is kind of based on exposure, on whether people have been exposed to gluten at certain times in their life. It's also associated with an overall increase in autoimmune disorders worldwide. So I think that there's probably a link there for sure.
So you talked about this, and I'm jumping ahead, but you did show how gliadin can actually create some paracellular leakage, so to speak, or that. So I see a lot of my patients that will have celiac disease and then show up with another autoimmune disease.
Mm-hmm.
So the question is, is one of the reasons why we're seeing so much autoimmune disease like Crohn's and ulcerative colitis, could it be precipitated by an inflammatory process in the gut, beginning in the gut?
Mm-hmm.
And I just bring this up because maybe you're at the tip of the iceberg on possibly one of the causes of why we are developing so many autoimmune diseases. What's your theory on how we raise these crops, and how gluten has or the wheat has changed over the years from the amber waves of grain to short, stocky plants?
Well, in terms of the molecular profile of the gluten itself, I'm not as familiar with that research as probably some other more agronomy-focused individuals are. But in terms of actual food products, so what we end up seeing in bread and in pasta, the overall gluten content really hasn't changed over time.
Really? Okay.
Yeah.
That's actually something that I think I've told my patients wrong. I've told them that we're probably getting more gluten, which is one of the reasons why we can have that.
No, but I found it interesting though, because you did reference the alpha, beta, and omega different gliadin subunits. And then even from there, there's subunits of 1, 2, 3, and 4, mm-hmm. possibly. And as you said, I don't want to press you on this, but maybe the expression is just simply different from all of those different units over time, depending upon them being GMO or not GMO or hybrid, or who knows. It's hard to say.
Yeah, it's also difficult when considering whether there are differences based on those individual gliadins, because that proline X glutamine pattern shows up in all of them. Sure. Repeatedly. Yeah. And in some of my papers, I've focused on alpha-2 gliadin as one of these specific proteins. And that one just happens to have many overlapping epitopes in terms of recognition in celiac disease, but it's certainly not exclusive to just that one subunit.
Can you comment on these different subunits? And in your paper, you discuss the starting point of causing an inflammatory cascade with interleukin 15, I believe it was, mm-hmm. IL-15. Mm-hmm. Can you comment on that? And that's in the relationship for my gastroenterology colleagues to lymphocytes, because we always look for intraepithelial lymphocytes. Mm-hmm. And I think you showed the mechanism through IL-15.
Right. So in the celiac disease inflammatory cascade, we start with the recognition of gliadin by the CXCR3 receptor on intestinal epithelial cells. And that's one of the prevailing hypotheses in terms of pathogenesis, is that the CXCR3 receptor will stimulate the release of zonulin. Zonulin will then trigger that paracellular leakage that you mentioned earlier. And as the gliadin passes through the lamina propria, then we see this release of IL-15 from the intestinal epithelial cells. And that IL-15 is what's going to recruit those intraepithelial lymphocytes. And so that infiltration of those lymphocytes into the intestinal barrier is one of the hallmark one of the hallmark traits of celiac disease from the clinical perspective, as I'm sure you're very familiar with.
Okay.
Like Marsh scores, for example.
Well, let's back that up, because that is a key, everything you just said. But I want my colleagues to understand this, because as somebody who is a strong believer in intestinal permeability, AKA leaky gut on Google, but intestinal permeability, if I'm talking to my colleagues, leading to other things, let's walk it back. You discussed how Gliadin binds to CXCR3, which is fancy and everything, but dumb it down for me really quick. Gliadin leads to this, which leads to zonulin, which leads to this, so that I can tell my patients and my colleagues can tell their patients, this is the process of why I want you to avoid gluten, or to avoid gluten, to avoid Gliadin.
Mm-hmm. So I guess what you would tell patients is basically that their intestinal lining is extremely sensitive to this individual protein. And when they take in this protein, their body it's mounting a response, almost as if there is an invader, an immune response to this protein, if that is helpful.
And you showed it is helpful, because zonulin is something that I look for. I actually found a lab that can order that, so I can look and see, okay, I believe that you do have some intestinal permeability. We know that infection, bacterial overgrowth, and in talks that I've given, I say that high ingestion of gluten leads to increased zonulin. I didn't have a mechanism how it did, but now we realize that you just explained it. So it's like every time you dial it down a little further, you're like, no, I believe it more now. So I've been saying that for a long time.
Yes.
Yeah, that is really cool. So you in your science have shown that zonulin leads to intestinal permeability.
I was not the person to discover that. But there is I believe
you have the opportunity to own it right now. Nobody else has stuck their flag in it.
I'm not going to take credit for.
You can take credit for everything you want until somebody else calls in right now and says, "That's mine."
Yeah. Brilliant man. There's a mucosal immunology lab at Massachusetts General Hospital. Dr. Alessio Fasano. Spearheaded that work. Yeah, I would love to take credit for it, but I think that would follow my career in a negative way.
Alessio Fasano, I've been chasing. I would fly and just listen to him lecture and then leave the whole rest of the lecture. Yeah, Alessio Fasano is one of my heroes. So yeah, for sure. That's why I thought it was funny. If you claimed it, I'm like, yes. He seems to have relaxed a little bit over the years. He needs to get back at it. That is awesome. Let's talk a little bit about the whole section that you have on treatments, because you did a great job of summarizing different treatments that pharmaceutical companies, different people have tried to do. I'm very keen to, well, anyways, I'll let you get into it, because you did a great job of reviewing all of it and to show what has promise, what doesn't, what's there, what has failed in clinical trials, before we even begin to discuss the polyphenols, because then the rest of your paper is about, I believe that this is probably the best mechanism. So do you remember some of the stuff that you have in the paper about a few of the different mechanisms that different pharmaceutical companies are looking at?
Yeah, for sure. So they can kind of be broken down into two major classes. So I think of them really as patient-focused versus protein-focused. And the patient-focused therapies are going to be kind of your immunomodulators, things that are affecting the individual's immune response, that inflammatory cascade through, for example, Nexvax was a vaccine that was, I think it got to phase 2 trials and then eventually was shut down. We have larazotide acetate, which is a zonulin inhibitor. That one is in, I believe, phase 3 trials now, which is pretty exciting. And those that target the immune response, I have to mention my favorite, which is the hookworms. So hookworm infection can actually mediate immune responses and take down what would be this overactive immune response in celiac disease. Immunosuppressive effects of hookworms have been.
So we have looked at trying to use worms for Crohn's disease for the same reason. How does it do it in celiac disease?
So in celiac disease, it just dampens the immune response. And what they've shown is that in vivo, individuals who've been infected with hookworms have a decreased IL-15 release upon stimulation with gluten. So.
Interesting.
That's the thing where we always say that in third world countries, you don't see autoimmune diseases. And a lot of us argued that point as to why. So.
Yeah, so that's probably my favorite that I haven't done. But then we have the protein-focused therapies, which I think the most notable are the enzymes. So if you're orally taking in an enzyme that will help break down gluten further, that's one that's been shown to be effective. Also some antibodies that will bind to gluten and prevent its digestion. So it's kind of a similar mechanism to what I talk about with polyphenols. And those protein focused are basically going to either break down or further sequester gluten from digestion.
Nice.
Yeah. You got into a little bit about how these proteins bind and how there's different mechanisms with polyphenols. Really quick, the Nexvax, was that Immusan T? Do you know the company that was doing it?
Yes. Yeah.
So years ago, Bob Anderson is a PhD gastroenterologist out of Australia. I had hooked up with him when we were just beginning the whole concept of using polyphenols to treat IBS. Mm-hmm. And it shows how difficult it is to really take a concept and take it all the way through to something you can get through the FDA, because it was like 8-10 years ago that he was working on this and it started a company. And then when you said that, I was reading your paper, I was like, oh, it didn't work. I mean, that guy put his heart and soul into it. And so shout out to Dr. Anderson, who really kind of, I think hopefully his next version will be able to do this so we can have a vaccine. Everybody's talking vaccines right now. So I'd love to have the old vaccine. So that's awesome. You want to ask anything about that?
About all of that?
About all of it.
Yeah.
We covered a lot.
Yeah. I mean, man, you must be tired. Thanks, Ken. That was awesome. No, I find it incredibly interesting the way that you've had this pathway to figure out what proteins are the biggest threat. I was surprised from what you had written specifically about hookworms, and I was just thinking about those who wouldn't have an inflammatory issue because they weren't wearing shoes. I mean, isn't that how you transmit hookworms? Is through the soles of the feet?
I do not remember my parasitology right now. Do you know, Charlene?
I'm not sure. I'm pretty sure they were orally administered, at least in the study. So.
Oh yeah, no. I'm sure in the study they were. My version was the old version.
All right. We're going to see if hookworms work here. Everybody take your shoes off.
Yeah.
That vat right there has placebos, or that vat has hookworms. Just jump around and see what happens.
So remember, before we recorded and started this episode, we told you we leave all of the mistakes in. That joke didn't work. And that's fine. It happens occasionally.
But I think it's really important because leading up to that, you're discussing all these different ways that pharmaceutical companies are trying to do it, and people are trying to figure it out using some really novel ways, like the hookworms, oral ingestion.
Yeah, not oral, not soles of the feet.
Not soles of the feet, but I think you're right. I do think hookworms happen because you get exposed to it. So then this is when you propose why polyphenols. So now this is the peeling back where now you're going to discuss polyphenols. So looking at all those different mechanisms, why do you 30,000 foot view, why do you think polyphenols will be beneficial? And then we'll dial down to the different steps. You've got some great charts and steps A through F on how it works and some incredible science on everything. So why did you think that polyphenols would do this in the very, very, very beginning?
So in the very beginning, it seemed like there was probably a pretty good chance that polyphenols would be able to be beneficial because we knew from those studies with using gluten as a fining agent in wine that there would be an interaction between gluten and these polyphenols. And then we have sort of this added benefit of knowing that polyphenols are generally safe. You know, people consume them every day as long as they're eating fruits and vegetables. And we know that they have these other beneficial effects. So for example, they have these antioxidant, anti-inflammatory effects that are occurring when these compounds are not interacting with protein. So we know that they're already generally a pretty good thing. And protein polyphenol interactions are just kind of this natural phenomenon that we see all the time in many different contexts. So just starting out, it seemed like it was probably going to be a slam dunk.
That's awesome. All right. Now, where was it? Page, for this, page 19. That little picture sums up a lot of important things. That little picture right there sums up about 15 or 12 pages of material. So this is your graph here shows stepwise why you can demonstrate how and then we'll get into this after you go through this, but a large polyphenolic compound with more branches can do some of these things. Mm-hmm. So this is kind of the steps A through F on how these polyphenols may help us deal with gliadin. So on your first part right here, can you please explain the physical sequestration of native gliadins?
So with native gliadins, that's going to be your gliadin before it interacts with any of your digestive enzymes in your gastrointestinal tract. So that would be the form of gliadin that's going to show up in, for example, a slice of bread. So these are going to be higher molecular weight proteins that are fully intact and haven't been digested at all. And these are able to interact with polyphenols through a variety of molecular mechanisms. So we see a lot of hydrogen bonding, we see ring stacking. One of my earlier papers really gets into the weeds in terms of the types of bonds that are formed. But we see these interactions and the formation of this precipitate in solution. So if you were to combine a solution of polyphenols and a solution of gliadin, you would see it actually form a solid and start to fall out of solution, which is a pretty good indicator that there is a physical interaction going on. So that's kind of the first step. And we see that also with gliadins that have been digested by pepsin and trypsin, which are two digestive enzymes.
Sure. What polyphenol did you start with specifically? What type of polyphenols?
So I specifically at first was interested in epigallocatechin gallate, which is the primary catechin found in green tea. Mm-hmm. I was interested in EGCG because of its prevalence in green tea, and we know that it has all of these other beneficial effects. It was being studied in terms of its anti-obesity and anti-inflammatory effects by another group at Penn State at the time. So it seemed like the one to target. And some of my later, more applied work used green tea extract. So that would also combine other types of catechins and smaller phenolic compounds.
Nice.
We're going to get into that in a little bit here. Yes, all right. Then in your beautiful little graph here, step 2, hydrolyzed gliadins.
Yeah, so what I just mentioned, hydrolyzed gliadins, that would be those which have encountered pepsin and trypsin. So breaking those proteins down into smaller digested fragments. And we see the same sort of precipitation forming, the same kind of haze formation when we combine those in solution.
So basically we're talking about a larger gliadin molecule versus a smaller one. Regardless, the polyphenol is going to suck up, grab it, and sink down to the bottom. What was that called in wine again?
Fining.
That's called fining.
Would you consider this fining also, even though it's not in the wine industry? Can we use it?
It is the same molecular mechanism, but fining, I think, refers more to the processing. But we do see protein and polyphenol interactions similar to this in a lot of different areas of science. So again, in wine, because that's sort of the root of this project, we see interactions between small proline-rich proteins and polyphenols in astringency. So like if you take a sip of red wine and you kind of get that drying sensation on your tongue, that's actually the polyphenols from the wine precipitating your salivary proteins, which are also rich in proline.
So that was, I always wondered that also. So, okay, so quick question, which is interesting. So if it, well, I'll save it for later because I want to go through the mechanisms first. So that's 2 mechanisms, but I got so many questions for you related to all this stuff.
Okay.
Then there's the aspect of digestive enzyme inhibition.
Mm-hmm. Right. So this is actually, I think, one of the really interesting parts of this study is polyphenols have been noted for anti-nutritional properties and the prevention of protein digestion in a few different studies in the past, showing that individuals with a high intake of polyphenols actually can sometimes have issues with protein absorption. So one of the reasons is the direct interactions with protein, like we just mentioned, between the polyphenols and gliadin. But another is the fact that these polyphenols can directly inhibit the action of digestive enzymes through either binding directly to the enzyme and kind of changing the shape of the enzyme, preventing it from being able to hydrolyze its substrate, or binding in that same binding pocket for the enzyme and preventing the substrate from even getting in.
So that's fascinating. So there is a little bit of a debatable thing, because when working with scientists in the cattle industry, there's actually some data to show that when you give sheep and cattle large like tannins, they actually have improved nutrition. But they're ruminants, and then people try and extrapolate the ruminants over here. And then we've worked with some scientists that have actually said, well, the binding, if it binds to the protein, not necessarily the protease or the lipase or whatever pancreatic enzyme it is, that when it gets, since the actual large tannin does not get absorbed, it stays intraluminal, the body can digest the protein off, and then the tannin keeps going. So it's interesting because I think that there is some debate a little bit where, like, you have people like Ann Hagerman that are talking about how it can be an anti-nutritional thing. And then you've got these other people in sports nutrition saying, no, actually, you can improve the overall nutritional value of foods by taking it with a polyphenol. And so it's a fascinating area. And I think that it's kind of a moving target. We have seen in my clinical practice and in the studies that we've done, we have seen beneficial results using polyphenols. And I think that's why it can do some different things, like work as a fasting mimetic molecule and things like that. That's really interesting. I did not know that it has the potential to bind to the digestive enzyme. I had not come across that before. Wild. So
yeah, because enzymes are proteins. So
it's true. Yeah. The next one, improve barrier integrity and transport, and decrease transport.
So there have been some studies that have shown in individuals with leaky gut or intestinal permeability that polyphenols are able to just directly affect intestinal barrier function. And I showed in one of my studies, actually in our untreated controls, that we saw an increase in barrier integrity when we just used green tea extract compared to the control. So in the absence of gliadin, we still saw an increase in transepithelial electrical resistance.
So improved leaky gut. Do you have a mechanism for that when it's not related to gliadin?
So in terms of improving it when it's not related to gliadin, it's most likely just related to the inflammatory cascade. And, you know, the gut is kind of just always in flux and always producing, you know, interleukin 6, 8, 10, just everything. And kind of mediating that response, I think, can expose potentially some underlying permeability that it can then reverse.
I love that you say that, because we talk a lot about other things that create intestinal inflammation, like high fructose corn syrup, polyunsaturated fatty acids like soybean oil and things like that, that have been shown to create an inflammatory response. So you said something really interesting. We're talking about gliadin here, but this also shows that it can help with the leaky gut, intestinal permeability, whatever you want to say, when it doesn't even, it isn't related to gliadin. It can actually do that, related to the inflammatory cascade. And we talk a lot about inflammation on this show all the time. Inflammation's good when you need it, bad when it stays there all the time. And unfortunately, our diets, I think that we tend to keep it inflamed. If you eat the typical, American diet, the SAD diet, you're gonna have an inflamed gut. You need to protect it.
Definitely. And you haven't gotten to zonulin yet, have you?
We talked about zonulin a little earlier.
No, I didn't know, like during the Cascade that you had written down with your hand. The reason why I say that is because you had long before, in conjunction with the leaky gut protection, you had mentioned that zonulin contributes to leaky gut. And since we had mentioned it earlier and you hadn't put it on there, that probably controlling or keeping the decrease of zonulin after having enough polyphenols would also lend to.
Well, now that he brings it up, so you wrote that Gliadin binds to MYD88, which ultimately releases the zonulin. Mm-hmm. So does it prevent the binding to the MYD88? Is that how it prevents the zonulin from being released?
So we don't know the precise mechanism in terms of what interactions it's actually preventing. So where it's really targeting that inflammatory cascade. It would make sense based on our data that the polyphenols are sequestering the gliadin and preventing it from binding to anything else. But in terms of studies where we actually pinpoint that mechanism, those haven't been done yet. So I think it would be really interesting to do some of this either in vitro or ex vivo if we were able to get tissue cultures to see how that affects really the zonulin release, because that's kind of the linchpin in that mechanism.
So that's fascinating. So I've had, I treat a lot of small intestinal bacterial overgrowth, SIBO patients, and I will check them and they'll have increased levels of zonulin. I assume it's related to the bacterial inflammatory cascade. Then we treat it and it goes down, treat them with Atrantil and it goes down. And I'm assuming I got rid of the bacteria, but you just brought up something interesting. There may be something more nuanced going on.
Mm-hmm.
Because most of these people,
really,
yeah,
a lot of different options.
Yeah, so I think that's another angle. It just shows how much, I think there's so much cool chemistry, cool physiology, cool applications of this that you're really tapping into, and you're bringing up your own questions. You're gonna have a long career chasing your own thoughts. So
hopefully.
And then we already talked about the anti-inflammatory activity, NF-kappa beta and things like that you basically discussed, which is the overall decreasing of the inflammatory response. And then the final one that you discuss in this figure is the transglutaminase downregulation. Can you explain that? That one I was struggling with.
Yeah, so that actually is from a really recent paper by a group out of Louisiana State University. And they found that when treating intestinal cells in vitro with cocoa polyphenols, they saw a downregulation of tissue transglutaminase. So when we have this exposure of gliadin to our intestinal cells, transglutaminase is released kind of as a response to epithelial damage. It's an enzyme that is found in everyone's body that's associated with wound healing. So when the when your intestinal barrier is kind of infiltrated by intestinal lymphocytes, sorry. When you see that infiltration and that damage that's occurring, that's when transglutaminase is released. And what they found in vitro was that we're not seeing that release of transglutaminase when polyphenols were added to that culture.
That's super cool.
You probably don't have the direct MOA on that yet, but it's definitely been noticed as a decrease. Is that what you're saying?
Right. Yeah.
Okay. No, that's very, very cool though. I don't think I'd ever heard that before at all.
No, that is really cool. And the mechanisms, how you go into all of this is just absolutely incredible. All right. So let's dumb it down real quick for me. Binding to my proline in my saliva. And now I understand why. So can you tell me how fat? So like if I eat a ribeye and then take a big, bold Cabernet, why the Cabernet doesn't have that astringency while I'm eating a fat? I've always wondered what.
Fat. Yeah, so fat will coat your tongue. And it's basically creating a layer between the proteins that are in your saliva and inhibiting that binding.
So it's making the tannin just slide over the proline, is what you're saying.
Yeah, you could explain it that way.
Well, I'm going to explain it that way to myself. I'm not going to get more complex. I'm just going to go, you're doing this to let it slide over.
I like the way she said, you can do that.
All right. So in theory, we have done, I mean, it's not fining because it's but you need to come up with your own term when polyphenols bind gliadin and sink to the bottom. So shining, Charlene, fining.
Yeah, it's Charlening.
It's Charlening. Okay. So when you're Charlening gliadin down in there, and then if I take, if I put a bunch of Gliadin in this cup, and then I put a bunch of polyphenols in there, and it Charlenings it down. Charlenings, can I turn it into a verb? Or is
Yeah, you did.
Charlening?
You just did.
So when I'm Charlening the Gliadin to the bottom of the cup, then I mix it up and I swallow it. This is the other concept I'm trying to think of. Does the body then, it goes down, I don't feel the astringency because it's already bound to the Gliadin or whatever. I'm not getting that, but will the body then break off? Will the enzymes, since the polyphenol's tied to the Gliadin, will my body break off that Charlening molecule? In other words, can it still digest the protein that is attached to it?
So in terms of whether it can still digest the protein, our data says that it will not. So doing an in vitro digestion of polyphenols and gliadins that have been sort of pre-combined before all of the pH fluctuations, before the introduction of pepsin and trypsin, we've found that complexation will prevent the digestion of gliadins, of native gliadins, and it will prevent the formation of those smaller molecular weight fragments that stimulate the inflammatory response.
So since it binds so strongly to the proline glutamine aspect, if you have a protein that doesn't have that, do you think that there will be less affinity and it will come apart?
Well, that's definitely something we're interested in looking at now that I have some grad students working on this project. We're really interested in seeing how kind of the matrix effect of everything else in the gut can affect these interactions and affect the stability. So one thing that I think suggests that we'll still see some success with this nutraceutical treatment is the fact that polyphenols generally have a greater affinity to bind to proteins that have a high frequency of proline. And the high frequency of proline that we see in gliadin is fairly unique. We don't really see that much in other food proteins. So it's something we'll need to investigate, but there's definitely a chance.
What are some other high proline foods that we typically eat?
High proline foods that we eat. I think you got me on that one. But in terms of the kind of major food proteins that we study in the lab, so I remember doing some sort of preliminary work with like beta-lactoglobulin and some casein when I was in grad school, just to kind of look at precipitation. Those definitely have less proline than gliadins do. In terms of others that have high amounts of proline, I'd have to look into that.
I remember looking once as to why there's a lot of soy issues, and I did see that soy has high proline compared to other proteins, and so I'm wondering if that's one of the reasons why a lot of people have issues with soy. Also, I don't know.
That's definitely something that's worth looking into because it is the high proline content that kind of gives gliadin that unraveled structure that makes it difficult to break down enzymatically. That's definitely something to look at, but not something I've heard or read.
Yeah. You've been slacking, Charlene.
Poor CV's only 17 pages long.
Yeah, come on.
Do something.
You sit there, you you come up with the term Charlening and you're just resting on your laurels. Just
Yeah, I know.
making money off that trademark.
Got to my head. So.
All right. Now, my favorite part of this whole thing. You said in your article, studies into mechanisms have shown phenolics with large branching structures with greater potential interaction have greater affinity for interacting with gliadin. So that's something that a lot of people don't really understand, is that these polyphenolic compounds come in varying shapes and sizes. I'm setting this up because the scientists that we've been working with in South America, the Quebracho, Colorado is one of the largest stable polyphenols that the original research I was doing is because it's both acid stable and it's basic stable, which means it can stay intraluminal in the intestine. And then the scientists we've worked with, one of them in an in vitro digestion showed that when this becomes fermented, in other words, with our microbiome, that it actually, she did a gas chromatograph and showed all these different molecules that came off, one of them being EGCG, another one being quercetin, another one being rutin. And you're like, holy cow, wait a minute. These large building blocks actually have all these smaller phenolic molecules in it. And all the studies have been done on smaller phenolic molecules because the prior studies were done on that. And you're going to grab a molecule that's already been done. Forgot his name, but the gastroenterologist that used EGCG to look at ulcerative colitis, and then that guy, because somebody else did it, and that's the proof of concept. What is really interesting is this whole science is using these large, if I gave you the largest, most hydroxyl bond, molecule, would this be the thing that would work better? I guess your paragraph implies that.
Yeah, so it's actually really interesting. And some studies that I've completed that we're kind of working on getting out there at the moment sort of take this idea of size versus actual shape. So when we talk about polyphenols and their structure, we're taking into consideration the molecular weight, but also the branching, like you mentioned. So if we compare molecules like theaflavin, which is found in black tea, versus EGCG, which is found in green tea, they have relatively comparable molecular weights, but EGCG has this hydroxylation that almost makes it from like arms. And it has this more flexible, bendable structure. And
I'm sorry, you lost me there. Would you show that one more time? What does it do?
It's awesome.
Yeah. So it kind of has these arms that are almost able to wrap around a protein, whereas theaflavin has, if you look at the structure, this benzotropolone ring, which is just this large, bulky sort of ring structure, and it doesn't have sort of that same flexibility as EGCG has. So even though theaflavin actually is a little bit larger, EGCG is able to bind better. So it really comes down to the structure dictating the function of the molecule, which kind of brings it to my lab and my fascination with all of this, the food structure and function lab.
Wow.
So yeah, figuring out how structure can affect the function of these molecules.
That's why you called it that, food structure and function.
Yeah.
Your website. I was like, that's an odd name for a website, foodstructureandfunction.com. And now it makes sense, because it's not just the food or the structure, but it happens to be that the two combine to make a function.
Right. Yeah. So structure and function are really interrelated in biochemistry. And
food structure function lab. Sorry, I just want to make sure everybody hears that. Foodstructurefunctionlab.com.
Structure and function lab.
Okay, just say it one more time.
Foodstructureandfunctionlab.com.
There we go. Food structure and function lab. Sorry to interrupt. I just want to make sure that if people want to.
Oh, no, you're fine. But yeah, conceptually, that is something that I just think is so fascinating, how the structure can really affect the function in terms of health. So as a butt doctor, the thing that's going to take that all up to another level is how the microbiome is involved with this. Then we're like, everything you're doing is fascinating and so cool. And then you start fermenting it and seeing what happens, and some pretty incredible magic happens. So right. Yeah. So fermentation, but then some of the work that I did during my postdoc at Rutgers University, we were looking at alternative mechanisms for polyphenols to affect the gut microbiome, because we saw with, I believe it was cranberry polyphenols as well as grapeseed extract, someone before me found that supplementation of those polyphenols to an individual with diabetes resulted in the increase of this growth of Akkermansia mucinophilia. And so we're trying to look at how why this happens, like what is the mechanism? And so one of the things that we really got interested in was the potential for polyphenols to act as radical scavengers in the lumen. So whether they're being broken down by the gut bacteria, whether they're scavenging radicals, whether they have an antibiotic capacity. I mean, there are so many options and it's so fascinating. So yeah. Sorry to go off on that tangent.
That's not a tangent at all. That's pretty much the world I live in, because I want to see my goal. What we have seen through fecal studies, actually, is that when my patients take either a diet large in polyphenols or they supplement with atrantil, we have seen an increase in microbial diversity. Mm-hmm. And then once you have an increase in microbial diversity, you start having different species of bacteria which can do these beneficial things, like the anti-diabetic, anti-obesogenic, mm-hmm. and then you have the and then people are now looking into that, and that's sort of this whole new field of science called the postbiotics. Mm-hmm. It's like, so we're almost describing that these become prebiotic-like, where mm-hmm. just something that is not digested, and then your own microbiome. But what we're seeing is that it's not a one-to-one. You have to have a diverse microbiome to get the full benefit of these polyphenols.
Right.
So you can have the structure and function, but you also have to have the ability to produce these postbiotics. Like, have you ever heard of a molecule called urolithin A and urolithin B? Things like that a lactic acid gets, kicks off, and we know that helps, with apoptosis and mitophagy. And there's a lot of stuff to uncover. This is a lot of really cool things. What do you think? All right, so just sum this up. Sorry about that. We covered so much material. So sum up your whole paper here in just a couple sentences, because there's a lot that we just talked about, and I want to hear it in your words. How would you describe what we just talked about in just a couple sentences?
Sure. So overall, we are focusing on developing a nutraceutical approach to treating celiac disease via the natural phenomenon of protein polyphenol interactions and keeping gliadin from being digested and being recognized by the immune system.
Perfect.
She did it in one sentence, Ken.
Yeah, she did. Wow. All right. What's your lab going to do next? What are you guys working on?
We are continuing to pursue this project. It was kind of on a hiatus as I did my postdoc working on a different project. But now that I have my own lab, I have a couple of graduate students who are really targeting that sort of structure and function angle. We're looking at extracts from different polyphenol-rich foods for their ability to basically elicit these same effects that I observed with green tea extract. And we're also trying to kind of target those downstream mechanisms that I discuss in the paper. So what is the impact of polyphenol supplementation on, for example, the activity of tissue transglutaminase? What is the impact on the recognition of these proteins by antigen-presenting cells? I'm kind of trying to go downstream and sort of look at this, the potential of polyphenols from really every angle of celiac disease.
That's awesome. You said you have 2 graduate students working for you right now?
I actually have 4 right now. Yeah,
I have
perfect. 2 that are working. I have 2 that are working on the celiac disease project, and then 2 more that are working on another project based on protein polyphenol interactions. We're looking at using novel plant proteins as delivery systems of anthocyanins.
Oh, wow. That's awesome. Can you do me a huge favor and settle a debate between me and Eric? Can you just take 2 of those and have one of your graduate students swallow hookworms, have the other one just walk around on hookworms to see?
We haven't even had a hiatus to have this debate yet.
I have a student who, an undergrad who really wants to work in my lab, so I'll pitch that project to him one day.
or undergrad, doing anything to get into graduate.
This is your hookworm internship. Nice. Well, Dr. Charlene Van Buiten, thank you so much. PhD as Assistant Professor of Food Science and Human Nutrition at Colorado State University. I can't thank you enough for coming. I want to repeat your website, foodstructureandfunctionlab.com, correct?
Yes.
Everything will be listed in show notes. We'll have links to your study as well as your own personal website. And Ken, any closing remarks?
I've just set your social media hashtag. There's no way you're as smart as I am. Is that it? Because that's what I feel. Do you have any social media or anything? Do you want to get out there?
I'm on Twitter, CB Van Buiten, if anyone has questions or is interested. I don't share that much science on there, mostly insight to working in academia, but that's really all I'm on. So
if you want hard-hitting political opinions, go to her Twitter account. No, I'm just kidding. Not at all. Thank you so much. Hey, don't go anywhere once we wrap up the show, but Gut Check Project fans, that is going to be episode number 52. Be sure and check show notes so you can connect with Dr. Van Buiten. And again, if you have anyone in your life or yourself suffer from celiac disease, we think that this information could be of just huge, great consequence for you and your family.
100%. And this is why we do this. We get an opportunity to meet brilliant people like you. Thank you for taking the time to do this. And hopefully this will get somebody to call your lab. Hopefully we'll be able to do some sort of collab. I would love to collaborate with your lab.
Likewise.
We'll talk more about that, but maybe another scientist goes, oh, this is the little piece I was looking for. This is how we help each other as a community. I'm a clinician. You're a bench researcher. I'm going to apply whatever you tell me and see if it works. And just thank you for doing everything that you do.
Thanks so much for having me.
Absolutely. That's episode number 52. We'll see y'all next time. Like and share. See you next time.
Bye-bye.